Let's go ahead and get started. And if folks join later, that's okay too. But we'd love to get to as many questions as possible at the end here. So we'll start right away. First of all, thank you all for tuning in to today's webinar. Today, we're extremely excited to be hearing from Dr. Matthew Harms about how whole genome sequencing and a deeper understanding of genetics can help researchers and clinicians better identify more tailored treatments for individuals with ALS. Of course, thank you to the rest of my team for working behind the scenes to make this event happen. And a special welcome and thank you to our speaker, Dr. Harms. I'm really excited to talk to you. And I'll introduce you shortly here. Just a little bit of background for those of you who haven't been to our previous virtual events. My name is Tanner Dagdalen. I am the co-founder and chief executive officer of Unite Genomics. And through the technologies that we're developing at Unite, we aim to rapidly increase the research community's understanding of ALS, get more patients into trials faster, and cut years off of the discovery cycle time for researchers. So for us, we envision a world where all patients are empowered to improve their health and no patient's disease goes untreated. And as far as we're concerned, this world is 100% possible. And as a community, we can make it happen. So a little bit about what to expect from this event. So Dr. Harms will first be presenting for about 40 to 45 minutes about the current research underway regarding his multiple ongoing research projects. And then after that, the two of us will have a follow-up discussion and answer as many of your questions as possible. If you have a question at any time during the event, please use the Q&A feature to submit your questions, and we'll answer them at the end of the session. I'm gonna look and see if the Q&A feature seems to be working. And it looks like it may not be. Okay, we're gonna do a game time audible here. So instead of using the Q&A feature, if you have a question, please use the chat to submit your question so that we can see it come in. We've been having issues with the Q&A box, and so we wanna make sure that we actually see your questions. Okay, so now without further ado, let me introduce our amazing speaker. Dr. Matthew Harms is an associate professor of neurology at Columbia, where he is also a principal investigator of the Harms Lab at the Institute for Genomic Medicine. The Harms Lab is focused on harnessing emerging genetic technologies to understand the causes of inherited neuromuscular diseases, particularly motor neuron disorders like ALS. So Dr. Harms, thank you for joining us today. If you're ready, you can go ahead and get started.
Yeah, thanks, Tanner, and the entire Unite Genomics team for inviting me to give this talk today. And over the course of the next 40 or 45 minutes, I want to basically try to break down the genetic causations of ALS and try to make what turns out to be a very complex emerging story as simple as possible. So hopefully I won't lose many of you, and we'll bring you along as we elucidate how it is that whole genome sequencing, whole exome sequencing, and other genetic technologies are allowing us to make inroads in understanding this really frustrating disease. So before I go on, I should just disclose that I have provided advice and insight to different companies who do genetic disease therapy development, as well as discovering new genes, and have multiple projects that are funded by some of these same organizations. I'll let you know if I think that that's going to conflict with anything that I shared today. So as many of you know, ALS gene discovery has been robust over the last 25 years. The first gene, SOD1, was discovered the year I graduated from high school, and things have really taken off from there. And you can see, according to this diagram, that essentially, we're at a pace now where 2 or 3 genes that are causative for ALS are being discovered each year. This is unprecedented and has given us a dramatic look into the underpinnings of ALS that is yielding fruitful therapeutic ideas, at least at this point. So I want to briefly review the human genome. This will be quite brief, including how mutations in DNA can lead to disease. I'll provide an overview of the history of ALS gene discovery and underscore basically how different emerging technologies have enabled these different discoveries. And then I'll talk briefly about how this is teaching us about the causation of ALS and where that will lead in the years to come. So of course, the human genome is 6 billion nucleotides long in each of us. It's made out of DNA and packed into the nuclei of each of our cells. And the cells, of course, use these parts of DNA as the code or the blueprint for creating the proteins. And you can think of each of the proteins in our bodies as a widget or a gear in a really complicated machine. And so if any of those widgets don't work or aren't present, of course, that portion of the machine isn't going to function the way that it's supposed to. Each of our genes, and we have now around 19,000 of them, the human genome was basically just recently completed with a whole bunch of little repair segments that tells us the number is like literally just under 19,000. But from these 19,000 genes, more than 80,000 different proteins or those widgets or building blocks are created. And as I mentioned, the proper function is key. And in fact, around 6,000 of these genes are already known to be disrupted in human disease. If you create a mutation in one of those genes, it leads to the broken building block, and that goes on to cause any number of types of diseases, including ALS. How do gene mutations work? Well, this shows two chromosomes, a chromosome that came from mom and a chromosome that came from dad. And on the DNA here are two different genes, just represented by the CHOMP region and the linker region in orange and green, respectively. When a cell needs CHOMP protein or linker protein, it goes to the DNA and it will make a copy that will serve as the blueprint. So it's going to make a copy from both mom and dad's chromosomes. So you'll get 2 RNA copies of CHOMP. And when the cell then uses those blueprints to create the protein, it will string together amino acids to create the protein, and it will take on the shape of the job or function that needs to be carried out. So, jokingly, this is called the CHOMP protein. And so it's shaped a little bit like a Pac-Man, and its job is to basically take things in the cell that look like a rod and to split them up into small pieces so that they're split in half. So similarly, if you need linker protein, you're going to create copies of the linker RNA, which is going to serve as the blueprint for translation, and create a protein that looks like it's supposed to link things. So in this case, maybe this blue and this red other proteins that need to get close to one another to do their function. And so linker brings them together. So you can imagine just how mutations, if they disrupted how these linker proteins or how these CHOMP proteins are shaped, that they might not do their function. So just to illustrate that, if you now introduce a mutation into mom's copy of CHOMP, you'll end up with one good copy of the RNA, which will lead to normal CHOMP protein. And you'll end up with one mutated copy of CHOMP, which when translated will lead to something that doesn't fold right, that has a misshapen jaw, for example. And now when the CHOMP tries to happen, it's dysfunctional. And so you get an accumulation of those rods that were supposed to be chomped up in the cell. And you can imagine that clumping up of those rods would be bad for a cell and might lead it to go on to degenerate. Similarly, if you have a mutation on the linker protein, you'll end up with a bad copy of the linker RNA, which will lead to a badly shaped linker protein. And now when these two things try to get close to one another, the blue one can't stick. And so it just drifts off, and now red and blue squares can't really work together to do their task. So you can envision how simple changes in the DNA can lead to really profound changes in the shapes of proteins that lead them to be unable to carry out the task for which they were designed. And if these mutations are disruptive enough, they can go on to cause any number of human diseases. So we have now uncovered probably close to 36 genes that look like they might be responsible for ALS. So why do we even bother looking for genes? Obviously, if you find one, that raises a lot of anxiety that it could have been passed on to loved ones or that other people in the family might be at risk. So why bother looking? Well, the first is that I know many of the patients I take care of are asking one basic question, which is, why did this happen to me? And so for the subset of patients in whom we can identify a gene mutation, this explains why they developed the disease. And also, I guess, gives kind of an enemy to be mad at, the gene mutation in your DNA. And that can be, a rallying cry to go after the SOD1 gene or the C9orf72 gene or what have you. When we look for genes in patients with ALS, we're also going to be able to figure out who else in the family might be at risk for developing the disease. most of these genes are inherited in what we call an autosomal dominant fashion, which means there's a 50-50 chance that children will have the mutation. And in most cases that one of your parents had it, and maybe without even knowing it. And so therefore your siblings might also be at risk. So knowing the gene also allows us to sort out who might be at risk in the future for developing the disease. Nowadays, there are technologies that allow us to use fancy genetic implantation and prevent embryos with mutations from giving it to the next generation. But of course, the reason why we're here today for this webinar is because what we really want to do is develop therapies that are specific for individuals who have mutations in these specific genes, and also learn about people who don't have mutations in the genes we know about today, to identify which pathways lead to neurons and neurons dying, and that will give us insight into what types of therapies might be useful for a broader group of patients, not just those with identifiable mutations today. That's exactly what's been happening over the last 25 years. I have assigned cellular functions to each of the proteins that have been identified mutated so far. And you start to see some emerging themes. You start to realize that RNA metabolism has many disrupted genes in ALS. Something called protein homeostasis also pops up. That's where cells are constantly clearing out the garbage. Proteins that misfold get degraded and recycled, but when that's disrupted, they build up inside the cell and cause the cell to die because there's just too much trash accumulating and mucking up the works. Other pathways that these different genes have told us about include the highways that go from the cell body of the neuron all the way out to the muscle. They're called axons. And the highways that things get shipped along can be disrupted in a process known as axonal transport. There are other pathways that are also emerging. And you can imagine that once we know what all of the pathways are that, when wrong, lead to neurons dying, you could start to design drugs that repair that or compensate for these big pathways, as opposed to targeting, let's say, the FUS gene or the optineurin gene. You would target the pathway instead and have a broader implication for the therapy you develop. So how we got about finding these genes, and how are we going to go about finding more of them? So the first era of gene discovery for ALS occurred from 1980 to 2000, essentially. This was before the human genome was sequenced and published, and it had several requirements. The first of these was that you needed massive families with lots of affected individuals who had ALS and had given DNA to researchers. And so basically, there weren't that many of those families around, but the techniques were utilized. You would basically identify the region of the genome that was affected, and then you would look across every base pair in that region to figure out where the mutation was. And some successes from that era, they were pretty good and helped us discover most of the very common ALS genes, just a few of which are listed here. So that was the first era of gene hunting for ALS. And using just those genes, we were one of the first groups to be able to use very cheap sequencing technologies from era 2 of ALS gene discovery to try to figure out how common these genes are. So just so we're all on the same page, I use the terms familial ALS and simplex ALS, or idiopathic ALS. So I put sporadic in quotes and familial in quotes because the line between these two is very blurry these days, because we now know that individuals with so-called sporadic ALS have a 1 in 10 chance of having a gene mutation that we usually find in people who have familial ALS. Now, the chance of finding a mutation in familial ALS is far higher. Many, many more people who have a family history of disease test positive for ALS genes. Now this is a little out of date. This study was performed just 6 years ago, and at that point, we were able to identify gene abnormalities in around 60% of families with ALS. But we could also identify mutations in those same genes in idiopathic patients, patients with no family history, in about 1 in 10. Now, this was around 500 patients just 6 years ago. The new technologies, next generation sequencing, the types of things that that genomic companies are now focusing on, make it a lot cheaper and easier to sequence folks. So I've now just recently completed analyzing 10 times that many patients, 5,481. And these genes in these different categories basically sort out identically. We're still finding causative gene mutations in 1 in 10 idiopathic simplex patients with no family history, and somewhere between 50% and 2/3 of patients with a family history of ALS. So what about so-called sporadic disease? What could be going on there where in the 90% of sporadic ALS patients where we don't find a single causative gene mutation? So there are a couple of things that could be occurring. So what we're able to find and look at today is a major genetic problem. And that basically explains that 10% of sporadic ALS patients where we can find a mutation today, or that 2/3 of familial ALS patients, like one single mutation that's bad enough all by itself to trigger neurodegeneration. But in the other folks, it could be any combination of factors that lead to the neurons dying. So it could be that there are 3, 4, 5, 6, 12, we don't know the number yet, really minor genetic problems that all synergize and pile onto one another to basically make the neuron unable to cope. It could also be that out there, not discovered yet, are some environmental exposures that are so bad all by themselves that they could trigger the beginnings of ALS. We don't yet know of any environmental exposures that in and of themselves are strong enough to do that, but they very well could be out there. Or it could be, in parallel to a bunch of small genetic problems piling on one another, you could have a bunch of small environmental exposures piling on one another in order to trigger the disease. And I would argue that most likely, in the vast majority of people, it's going to be something more like a handful of genetic problems plus a handful of environmental exposures. And I want to pause here and say that one environmental exposure that is clearly playing a role in ALS is the aging process. Now, none of us can avoid that, so it's not really an exposure, but it's a risk factor. As we age, our cells get worse at coping with a bunch of genetic problems or a bunch of environmental exposures. And so it could be that age plus genetic problems plus environmental exposures are all working together to create ALS in most individuals. And of course, any one of these is going to do the same thing, which is to kind of kill off the neuron and give us what looks like ALS on the outside. So you could have this vast variety of underlying causes ranging from purely genetic to purely environmental. And from the outside, you couldn't see that. It would look exactly the same in my clinic because what I'm seeing in the clinic is the deterioration of the neurons and the weakness or the spasticity or the atrophy that it's bringing about. And so what we're really after in ALS these days is figuring out the biological underpinning, regardless of how it looks on the outside. So it could be that patients with onset in their swallowing muscles at the age of 80, some of those patients have less in common with one another biologically than with a 25-year-old who has onsets in their legs. The biological underpinning could be the same, but the way we see it in the clinic could be entirely different. And I think that poses a challenge for how we've been doing clinical trials, makes it very hard for us to figure out who to include and in which trials, and something that we're all working towards remedy. So I just want to go through how it is that one big giant mutation or a handful of small ones could lead to the beginnings of ALS. And I want to use kind of this balance to kind of illustrate this. So if the balance tips in the favor of the insults, then neurodegeneration and motor neuron loss is going to occur. So for what we call high penetrance mutations, those mutations that are bad enough all by themselves to cause ALS with no other additional factors, basically act like this. You've got this big giant bad mutation and it's like a massive weight just sitting on that insult side. And for many years, our bodies are smart. And so they pile on these compensations, but eventually they're just not enough. And the compensations don't outweigh the insult coming from that SOD1 mutation. And so it tilts in favor of motor neuron death. But this is, a handful of situations in ALS, maybe 15% of the time. And so we're also envisioning that there are what we call low penetrance mutations, maybe getting even close to the category that some people might call a risk factor. In this situation, you might have a mutation that's not so bad, like the SOD1 I114T or I113T mutation. So it's a much smaller weight on the insult side. And in many people, the compensations that the body puts up are good enough, and they prevent neurodegeneration. In fact, for SOD1 I114T, about half of people who carry the gene mutation eventually go on to express the disease. So for those individuals, those compensations are adequate. They do the job for the person's entire life. So there must be other factors that kind of weigh in and trigger neurodegeneration in the 50% of people who do go on to develop disease. So one of those, just for example, might be the fact that you are male and have only one X chromosome. And so it joins in with SOD1 I113T, and we know that men are more at risk for developing ALS, and maybe in some individuals that's just enough to tip the balance in favor of developing ALS. So just to be clear, I'm never in favor of developing ALS, but it tilts it towards ALS developing. But then there are also these risk factor mutations, ones that in and of themselves are too small and don't have enough of an effect to cause much of a problem, but together are bad. And so here, on maybe, 3 different genes are these tiny mutations that only weigh a small amount by themselves. And, these 3 balls weigh exactly the same as the compensatory balls that the body's able to pile on. So the compensations in the insults are perfectly weighted in almost everyone. And then one additional risk factor maybe piles on. Let's say a traumatic brain injury or a whiplash injury or something like that, that basically just injures a small region of the neurons. And maybe combining with those 3 insults, it's just now enough to tip it over and to cause ALS. So when we look at kind of how this plays out, we've got big bad mutations like the SOD1 A4V, and then we've got all of these minor mutations piling on one to another. And so when we look in the clinic at patients, what we're going to see is what looks like single gene causes on one end of the spectrum and multi-factors on the other end. And so you can imagine that we're going to, in the clinic, see these folks where the mutation by itself is enough as familial cases, and in where it's multiple piling on, those patients are going to appear like they have a sporadic or idiopathic disease. So ALS spans this gamut, and the technologies that we've had to date have really only allowed us to approach the far left half, those really big bad mutations and the mutations that are pretty bad. We're just now getting to the era where enough patients have been sequenced, where we're being able to start to generate what we call polygenic risk scores for this other end of the spectrum, where there are multiple mutations piling on. And so stay tuned, because those are going to become very powerful ways of us figuring out who's at risk for ALS and why they developed ALS, over the next 3 or 4 years. So back to the ALS gene discovery timeline. So we talked about era 1, which basically got us up to the blue here. Then we entered into era 2, which is kind of represented by the green dots. And these were where you could skip many of the onerous steps required for the first era by going to whole genome sequencing, or by using the whole genome sequence to figure out which regions to look at. And these are the genes that came along in that era. And then finally, now with whole genome, where you just sequence everything and look later and interpret later, we're also being able to identify these genes that are in red by what's called burden testing. So the current era of gene discovery is being driven by massive scale analysis of genomes. We're talking 2,000 to 20,000 genomes to try to identify these genes. So how many undiscovered ALS genes are there? So Tanner will probably find this more interesting than many other people, but a friend of mine did kind of an ecological survey methodology where you basically capture a bunch of animals in the woods and count up what different species there were, and then you capture them again. And based on how often you recapture the same bird, you can estimate what the biodiversity is. And so if you do that same kind of an analysis for the human genome, basically we come to the conclusion that there are greater than 100 ALS genes yet to be discovered. So we've got to keep going is one major theme to take away from this talk today. So how are we doing that? Well, we're using burden testing. So what's burden testing and how does it work? Okay, so believe me, by the end of this, you will get it, but just hang with me because it's pretty complicated. So you basically take as many genomes as you can find from people without ALS and as many genomes as you can get your hands on for people with it. And across the entire genome, you ask, are there places where there are differences in the genome from the normal genome that we think look benign and like they aren't going to cause a problem, or bad and they might cause a problem? So illustrated here are a bunch of different ways to do this. And the green stars are the places where our bioinformatic algorithms have assigned benign changes. And the red lightning bolts are the ones that basically are locations where it looks bad. Like this region should not have abnormalities, and there's an abnormality sitting right there and it looks bad. So damaging variants, mutations that are likely to disrupt the protein. And then you basically add up across all of those people without ALS and all of those patients who have ALS and look for statistical differences. And this will tell you regions of the genome where mutations cause ALS. And so the statistical methods don't matter, but they've gotten really refined over the years that take into consideration all sorts of variables, and it allows us to identify new genes. So we wanted to apply this all the way back in 2015 to see what we could learn from just 3,000 ALS cases and about 6,000 controls. So we put together this discovery cohort, the ALS Exome Consortium. And this was groundbreaking because we got all of these researchers to work together. We got them to realize that they weren't going to discover new genes on their own, but that we all had to work together. And this has become the model for how ALS genetics works even to this day. And by combining those 3,000 cases and those 6,000 controls and doing that burden test that I just showed you, we were able to rediscover old ALS genes. So if we had not known about SOD1, if we had not known about TAR-DBP, we would have discovered those statistically. And we also were able to discover two brand new genes, the TBK1 gene and the NEK1 gene, which went on to kind of found new pathways that I showed you previously. TBK1 told us that microglia and inflammation was playing a bigger role than we had appreciated previously. NEK1 has led to kind of its own offshoot in investigating the pathways that NEK1 is involved in. And so it's opened up new pathways and avenues for us to look for ALS gene targets. So that was good, but the genome is far more complicated than just genes. Like individual regions of the genes vary one to another. And there are regions outside of the genes that also play major roles in human health. And so we needed to come up with new methodologies that would take that same burden method that I showed you, look for bad stuff, look for good stuff, and compare the ratios between ALS patients and controls, but apply it outside of genes so that we could find additional regions. And so I won't bore you with the details because it does get kind of arcane, but we basically were the first to develop regional collapsing methods that use new ways to look at different regions of the genome, to parse them into different features, and basically use that information as opposed to the individual genes to try to identify regions that are associated with ALS. So this method works better for discovering ALS genes. So I'll just show you this diagram. So the higher the dot is away from this red line, the more robust and obvious it is as an ALS gene, essentially. And so when we did this the first time, we immediately started to discover new ALS genes that were coming off of the red line. With our larger cohorts that we've just analyzed, I can tell you that some of these are coming up quite nicely. And that happened whether we looked at just Europeans, which has been one major challenge in genetics that I want to return to in just a second. But we also learned that by using these new technology, these new techniques for parsing the genome, we could ignore people's ancestries, which allows us for the first time to be able to use the genomes of patients who are not European to expand and diversify and include patient subgroups no matter their geoancestry, in our ALS gene discovery, which has been a major problem, right? Most of the DNA discovery and most of the gene collection has focused on white people. And what we're learning is that there's much to be discovered, and it's super important for us to diversify those approaches. And looking at the genome in the way that we've designed allows us to do that, even while we're making efforts to collect and to include people who are not white in ALS research studies. So I just want to return to this theme that genes implicate the driver pathways. And if we could understand the biological pathways in each patient, we would have a better chance of treating them and carrying out what we call personalized medicine or precision medicine. So the more genes we learn about, we'll learn more about what neurons need for health. We're going to find unsuspected pathways that will lead us to new ideas for how to treat ALS. And the more genes we find, the more implicated some of these other pathways will become, and we'll realize that some of them are really super important and the others play a minor role and maybe shouldn't get all of our research funding. So the ultimate advance, though, in how we find these genes is more patients sequenced. So it's all about numbers. So we continue to recruit patients and continue to sequence genomes to try to drive additional gene discovery. That leads me to the GTAC study. I'll just briefly tell you about it. It's wrapped up now, but the idea was that we would pair very good clinical data as we followed people over 3 years with their genomics to try to learn more about ALS. So the sites were distributed all across the country. The investigators worked extremely hard to collect samples. And I was just telling Tanner that it cost us $7 million and took 5 years to get to roughly the same number of patients that over the course of opening the beta test platform at Unite have been able to get their hands on over the course of just a handful of months. So, you know, this was back in the day where you had to, anyway, I'll spare you the boring details, but we're at the cusp where basically online platforms and things like that will enable much faster achievement of those higher numbers that we're looking for. I also want to point out that we're not the only people doing this. So we're in close collaboration with these other 4 projects, which are also bringing in patients with whole genome sequencing. And I think between the 4 of us, we're now approaching close to 20,000 ALS genomes, which I think might yield 1 or maybe 1.5 additional ALS genes. The next raft of discoveries will come as we close the distance between 20,000 and 50,000, if you ask my honest opinion. But we're on our way there is the good news. So why continue the gene hunt? Well, we want to provide answers to those key questions. Why you and not someone else? And could this happen to the ones you love? We want to empower precision medicine, and we want to identify new gene targets. These new targets will be individual proteins, important cellular pathways, and then in the empowering precision medicine, we want to be able to stratify patients for the next generation of clinical trials, find responder groups for drugs that we already have, and eventually guide patients to the best choice once we have numerous drugs to choose from for ALS. So what's that look like? Well, you know, as you all know and are equally frustrated to me, I assume, is that we've had more than 60 phase 3 randomized clinical control trials, which have led to 2 FDA-approved medications. What's typical? Well, about 10% of drugs should be making it. And so by my calculations, that means ALS should have at least 6 by now. So why isn't that the case? Well, it's because drug-specific issues have existed. So some of the drugs didn't even cross the blood-brain barrier and get into neurons. Some of them, your kidneys were so good, patients' kidneys were so good at clearing them out that they never achieved high enough concentrations to have done anything. Sometimes the studies were designed with too few patients or didn't study patients for long enough to even detect a difference. And that's because of kind of optimism amongst investigators who thought that they were going to find a drug that, you know, stopped the disease in its tracks as opposed to just slowing it down. I think from my perspective, the main reason though is that there is a big difference in underlying causes across ALS. And if you do an all-comer study and just group people based on the diagnosis, you're unlikely to get at that biological underpinning. So what does a precision medicine informed clinical trial look like? Well, if these red humans all have ALS, on the outside, they just look like they have ALS, but undoubtedly, if we could just kind of peer beneath the surface and look at the neurons and see what's going wrong, we would be able to discern subgroups. People where, that are blue, green, yellow, and red. And then we would be able to figure out what those underlying causes are. And then if we can basically partition patients out into their underlying biological causes, that's what we call genetic stratification. If you can do that, the number of participants that you need for clinical trials goes way down. And so you can be more efficient and it's cheaper to run clinical trials. And you have a better chance of seeing the effect because you compare the underlying biological cause with the mechanism of the drug, right? So you would never choose patients with underlying problems with protein recycling if the drug was targeting neuroinflammation. You would target patients with neuroinflammation for the neuroinflammation drug. And the recycling drug that boosts clearance of misfolded proteins to the patients who have the underlying problem with protein recycling and protein misfolding, right? So you compare the group to the drug and obviously by that mechanism have a better chance to seeing an effect. So mainly what we're proposing is that, if you can figure out what those underlying pathways are, figure out which patients have those, then you could pair the right medication to the right patient for the clinical trials. And then eventually on a broader scale, patients come in, sequence their genome. We do other types of sequencing on their transcriptome, see what proteins are in their spinal fluid and be able to say, oh, hey, I recognize this pattern. This is a cellular transport problem based on what I'm seeing here. And now we have 2 FDA-approved medications that target cellular transport. So we'll give you those medications. Skip riluzole because you don't have a problem with glutamate toxicity. Skip X, Y, or Z because you don't have a problem with protein recycling and target just the cellular transport proteins to patients with the cellular transport problems. So folks, we're getting there. We're not there yet, right? We have to develop the biomarkers who tell us who's who. We have to develop the drugs that target those individual mechanisms. And actually, that part's kind of happening already. There are more than 50 drugs with well-defined mechanisms falling into these pathways that are either entering clinical trials or already in clinical trials. So now we just have to do the hard work of looking at the biofluids, identifying the biomarkers, identifying the genetic signatures that tell us who's most likely to respond. And 10 years ago, there were different groups working in their silos to try to figure all of this out and therefore having a low chance of making dramatic progress. And today, in some measure due to kind of everybody coming together for these big data projects, that's no longer the case. Everyone is collaborating, sharing data, validating their findings in one another's studies in a way that is essentially unprecedented in ALS. And so I'm very optimistic that that type of collaboration with the numbers that we're getting to in our genome sequencing and our biofluid biomarker discovery projects are going to yield the types of discoveries we need to make precision medicine a reality as opposed to just a future goal. So I'll pause there and we can go ahead and take some questions, and I'll stop my sharing so that it's easier to see everybody.
Awesome. Thank you so much, Dr. Harms. Amazing talk. If you have questions and you haven't submitted them yet, feel free to submit them in the chat box. Again, the Q&A feature seems to be having some technical issues. So if you have a question, post it in the chat. But I'll go ahead and get us kicked off here. I have lots and lots of questions. The first, I think, is, you mentioned that we generally know a certain number of genes that are causative for ALS. And we know generally what pathways they work on or they affect. But even if a drug isn't specifically targeting a gene, but it works on a particular pathway. So an example is like Amylyx's drug that they're in development on right now. It works on neuroinflammation. Is there still value in using genetics to select for patients with ALS with kind of known pathogenic variants in those specific pathways, or?
So yes and no, right? So I think the genetic information right now is going to be more informative for targeting antisense oligonucleotides and potential gene therapies, like basically things to take out, kind of those big bad mutations. So I think that understanding who has neuroinflammation and so there are some pathways that are likely to be active in most patients to varying degrees, right? So neuroinflammation is one of those. Everybody has some. The question is that really driving the disease in a given individual? And sorting that out can be challenging. We can image neuroinflammation in people with ALS in the brain. It's very challenging to do in the spinal cord because the spinal cord is really tiny and it's really hard to get the signal right. But you can image neuroinflammation and start to stratify like who has a lot of it and who doesn't. The challenge, I think, from my perspective is, what do you do with neuroinflammation? Do you skew it to a different type of neuroinflammation? Do you squash it all together? Do you try to ramp up some parts and turn down other parts? And so what do you do kind of with neuroinflammation is a particularly challenging one. The other thing I'll just say is, everyone from their preclinical studies thinks they know why the drug might be particularly effective in ALS, but it's really rare that our drugs are as clean as we think they are. So even Amylyx, revs up some aspects of mitochondrial function, has an effect on neuroinflammation, does some things with reactive oxygen species. So there are these kind of multiplicity of different ways that many of our drugs act, and so it can be maybe hard to target even at this point. So a lot of companies are collecting DNA and doing whole genome sequencing in parallel with their clinical trial. And then, coming to folks like me, having me analyze it and basically saying, can we predict who responded based on the genomic sequences? Like, do people who have the UNC13A variant that's relatively common respond better to a given drug compared to people who didn't have that mutation, and start to kind of look for these genomic influences on the outcome of a given study or a response to a particular drug.
So in those instances, generally the sample size is pretty small. So is it very challenging to be able to identify those sorts of drug response related effects?
Yes, absolutely. It is very difficult. So you have to have something that's relatively common in order to even have a chance. Something that's rare, that might occur once or twice in your cohort, or three times in your cohort, you're unlikely to be able to see an effect unless it's dramatic. Like if you had a study that happened to include, a handful of C9orf72 carriers, and, looking back, nobody had any change unless they had a C9 mutation, and the C9 patients got stronger, maybe in 3 people with an improvement, you could see something like that. But you're right, it is challenging on the numbers end to be able to see something there.
And, I'm seeing some questions about Biogen's ASO program. So, and just, before we get into it, I think, my background is in genomics as well, so I can kind of get into jargon without realizing, but I'm going to try to use as plain English as possible so that we can be as clear with the community as possible. And maybe I can just kind of ask some probing questions that will kind of paint a picture for the community about some of the gene therapies that are, or not gene therapies, but genetically targeted therapies that are in progress right now. But one thing before we get to that is, you mentioned that it's common for folks in the community to believe that you aren't likely to have a genetically driven form of ALS, like SOD1 ALS or something like that, unless you have a family history of ALS. But at least for SOD1, there's data showing that idiopathic SOD1 ALS is significant. And I think it was even Biogen who showed some data that there are actually more, or they mentioned casually, that there are more SOD1 patients that don't have a family history than those that do have a family history. So, can you kind of give an idea of what that might mean for the clinical use of therapies that are targeted for a particular genetic gene like
Sure.
SOD1 ALS,
Yep.
yeah, maybe you can I'll ask kind of follow-up questions as needed.
Yeah, absolutely. So the first thing is, Amar Al-Chalabi from London basically did this elegant paper where he charted ALS gene penetrance and the number of kids people have and the likelihood that they would appear to be sporadic or familial if you met them in clinic. And basically the way it turns out is that if a gene mutation is 70% penetrant, meaning if you have the gene mutation, 30% of people will escape or die before they manifest symptoms, and 70% of people will get symptoms. And only 2 kids per family, half of mutations will be in people with a family history that's obvious and you can tell, and the other half will be in the sporadic population. And so that's true for C9orf72, that's true for TDP43, that's true for basically every ALS gene. There are more people with no family history with the mutation than there are families running around with the mutation at any given time. Gotcha. So I think the first implication is that, we need to expand genetic testing to at least include those genes that have genetically directed therapies. So as soon as we learn that either the C9 or the SOD1 or the FUS or the ataxin-2 ASOs, any of these antisense oligonucleotides that are in clinical trials, as soon as we hear whether they work or not, if they work, that will be a strong rationale for basically universal screening of those particular genes in all ALS patients, regardless of family history. Now, there's a second kind of caveat there, and that is that, some of these genes are implicated kind of in other ways in non-genetic ALS, right? So ataxin-2 increases the amount of TDP43 that's dysfunctional. So potentially using the gene therapy that knocks down ataxin-2 could help people who don't have an identified gene mutation, and that's being explored. Some patients who don't have an SOD1 gene mutation still have misfolded SOD1 protein in their neurons when you look later. And so it might be that there's another group of people who don't have an SOD1 mutation, but who have misfolded SOD1, in whom knocking down SOD1 with the antisense oligonucleotide might be helpful. So I don't know if it certainly wasn't succinct, but hopefully.
And I think I can, we'll get to, I think what the community really cares about here is that, let's say the results are really positive and powerful, and we see some of these therapies make it to the market that are targeted for specific genetic variants. So let's just look at tofersen, which is a drug from Biogen that's targeting patients with mutations in SOD1. So if it's the case that, there are many different genetic variants that could show up in SOD1, right? And so if is it the case that the drug is likely going to work best in specific genetic variants to the extent that the specific variant that you have in SOD1 could potentially determine whether or not that drug is a good fit for the patient in question, or are kind of all SOD1 mutations likely to be treated the same, and you don't really need to go into extra detail? So
I think that it is likely that the response will, so I don't think that like, let's take SOD1, for example. I don't think that tofersen will be less effective at knocking down SOD1 depending on your gene mutation. It will be equally effective at decreasing the amount of SOD1 that's in your system. The question is, does that manifest in slowing down the disease the same across all of the different mutations? There are some SOD1 mutations that are very, very slowly progressive. And so the body, is by and large able to keep up. And it may be that knocking down the SOD1 is not slow down the disease, obviously, in those individuals, whereas those with the really fast progressing ALS, where basically like every misfolded molecule of SOD1 is just piling on and sticking and, that would have a more dramatic effect. So I think we'll get some preliminary information about that based on the different mutations that were in the tofersen study, when that data comes out.
And kind of a follow-up question about that I think will be really relevant once there is an approved therapy that's targeted for a particular gene or set of genes. How educated is the physician community in the genetics of ALS? Even if you just look at neurologists, do they understand the value of what you might be able to learn from genetic testing, or is there still a big kind of educational component that needs to be addressed as well?
So I think in the ALS community, it's pretty robust. Most ALS clinicians working at certified ALSA or in Muscular Dystrophy Association ALS clinics have had to be our own genetic counselors for so many years that we've kept up with the literature and are pretty good on that front. The average neurologist is unlikely to. But we've made a lot of inroads, and like I chair the Northeast ALS Association's frontotemporal dementia and familial ALS panel or subcommittee, and we regularly design patient education symposia like this where we go over the genes, where we talk about the most efficient way to test them. I'm working with a couple of investigators now to develop official clinical testing guidelines for ALS genes, so that laboratories and clinicians can look at a paper and just follow it along and be able to arrive at the right answer for a reasonable cost and without generating tons of anxiety over what we call variants of unknown significance, which is the downside of genetic sequencing is that you're always going to find something. The question is it relevant to your disease? And that can be a slightly harder question to answer. Yeah. But I think most ALS clinicians are sending genetic testing. Often, at least in our surveys, it looks like many are universally sending C9orf72 because of just how common it is. Others are starting to add in SOD1 because in anticipation of tofersen results, maybe to kind of know where all of the SOD1 carriers are, so that if it's successful, who to start doing the prior authorizations on. But I think people are sequencing a lot, and so that's a useful thing.
Yeah, and I think there's a kind of general question about going back and doing kind of retrospective analyses of past clinical trials. You mentioned a lot of trials now are including genomic sequencing. This is relatively new, considering the cost of sequencing has come down so quickly.
A lot, yeah.
Yeah. So, what do you expect big things to come from, after the fact retrospective analyses of past clinical trial data outside of looking at response to that particular therapy?
Right. So the short answer is that, the way drugs get developed is they're designed by one company, bought by another company, developed in collaboration with another company that then gets sold. And so by the time we've gone back to try to do genetic sequencing, those assets are locked up. Like there isn't a legal way to figure out who can give permission for the analysis of those genomes. So, in the previous era, I would say like for the last 4 or 5 years, that issue has been taken care of. But the really well-defined patients, that you would love to sequence are just out of bounds. And in fact, that's in some respects, a testament to the informed consent process that we all adhere by, right? Many patients back in the 90s and 2000s had DNA saved, but no one asked them if it was okay to sequence their genome and to share it with the world. And they might have said no, and we don't know. And so it would be kind of violating their autonomy in retrospect to go back and do that sequencing. But nowadays, the sequencing is happening. People are preparing to be able to do that. The New York Genome Center ALS consortium that I help lead has a pot of money set aside so that if a company can't afford to do the genome sequencing in their clinical trial, they can come to us. And if they're willing to share the clinical data on the patients, we'll sequence the genomes for free so that they can pair that genomic data with the clinical trial information for exactly that type of after the fact analysis. But Tanner, like you were kind of alluding to on the numbers game, clinical trials, are going to be 300 people or less in general. And that does limit the number of questions that you can successfully answer with genomes. in a single study. We're going to have to kind of accumulate a bunch of studies in order to be able to get more usable information out of that.
Mm-hmm. And I think one other thing to mention here is that generally speaking, again, there's a lot of attention on whole genome sequencing. And a lot of individuals may have whole genome sequencing results that were sequenced a year ago, 2 years ago, 3 years ago. We know more now than we knew back then. And so it's entirely possible that if you go back to the companies that did your sequencing in the first place, you may need to have a physician order a reanalysis. But if you go back to the company and ask them to reinterpret the data from your original sequencing, they may actually know more about the status of different genetic variants than they knew at the time, and can give you a slightly different answer. So I just want to mention that as a potential resource for anybody who does have genome sequencing data out there. And we have lots more questions, and I'll take a look through them and see if we can get some answered offline as well after the fact. But I do have one last question for you, and I think it's kind of overarching here, which is that you mentioned that the clinical presentation of ALS can be very different for two patients that may have the same driver mutation. And this is similar to how we used to think about cancers in terms of the location of the cancer. We used to think this is a lung cancer versus this is a pancreatic cancer versus this is a breast cancer. And we've since discovered that the driver mutation that's causing the cancer is more important than the location of the cancer. So, and that's led to a huge leap in the survival rate in certain cancer types that have a drug that's targeting, their driver mutations and the pathways affected by them. So I guess the overarching question is, do you expect that the story with ALS is going to be similar to what we saw in oncology, or are there significant differences?
Yeah. So I think it will be, thankfully we're kind of dealing with one cell type, or kind of a handful of cell types, so it won't be, I don't think it will be quite as complicated as it is in cancer. I also think that maybe, there's a distinction between driver mutations and kind of driver mechanism pathways. And, in these cancers, you see like one type of mutation in one gene that leads to this dysfunction and the drug targeted to that. I think we won't even need to get to the point where we identify the individual mutations that might be leading to that pathway showing up. But if we can just get to the pathway, then we'll be, halfway there to being able to identify the drug that has a higher chance of working in that particular patient.
Awesome. So, with that, I really appreciate the time, Dr. Harms. This has been fantastic. We did record this event, so we'll be sharing the recording out for anybody who happened to miss it, or if you want to go back and review what was discussed today. Thank you again for joining us, and again, a very special thank you to you, Dr. Harms. So for everyone who is here, who attended, look out for a post-event evaluation form so that we can get some feedback about what was useful and what worked really well and what didn't work in this webinar. We appreciate everyone's support and really look forward to what we can accomplish together as a community this year. With that, as always, the time to act is now. So, you heard a number of different possible opportunities to get involved and learn more about what might be, lying dormant in your genome. And, there are lots of resources out there to investigate it. So with that, Dr. Harms, is there anything else that you wanted to add at the very end?
Yeah, I was just going to say kind of one thing. You were encouraging people to get their genetic sequencing reanalyzed to see if there's something that was missed the first time around. I would say just as important is to have it reanalyzed to clean up some misconceptions. So for every new, abnormality that we now know causes ALS, I would argue that there are 3 that we thought caused ALS and now we know don't. So there also might be some people out there who had a positive genetic result. They've been living with this thought that an angiogenin mutation is the cause of their disease, or a FIG4 mutation is the cause of their disease, or a specific abnormality in the sequestosome 1 gene is the cause of their disease. And the field has moved on, and we now realize that that is just a variant, a mutation that's pretty common, way too common to cause disease. And you may not learn about that unless your sequencing gets reevaluated. So kind of another reason to have somebody take another look, because it may be that what you've been thinking was the etiology or the cause of your disease is not.
Awesome. Thank you so much, Dr. Harms. And thanks to everyone who joined us. Looking forward to seeing you again at future events. And hopefully we'll have Dr. Harms back for some updates here in the future as well.
You bet.
All right. Thanks, everyone.